Hi there, my name is Carly Britt, and I'll be talking about DNA structure. To do this, we need to break the DNA down, so in your mind, kind of tear it apart, and we'll start from there. Not that kind of breakdown. So DNA stands for deoxyribonucleic acid, which means that it has three components: a nitrogenous base, a deoxyribose sugar, and a phosphate group. DNA also has an inherent grooviness, which we'll talk about, and a handedness, which we will also discuss. So the four bases in DNA are adenine, guanine, cytosine, and thymine, uracil, and RNA. These bases have two structures. So A and G are your purines, T and C are your pyrimidines, and there are base pairing rules here. So adenine pairs the thymine with 2 hydrogen bonds, and guanine pairs the cytosine with 3 hydrogen bonds. The bases themselves are hydrophobic. Hydrogen bonding happens just between the bases, but remember the bases themselves are hydrophobic, and this impacts base stacking later on. So we have a deoxyribose sugar in DNA, which just means that it's missing an oxygen on that second carbon. And so we count these carbons so that we know where each connection is happening. So on that carbon 1, you'll have your base, and then your 5' carbon will have your phosphate group, and with your base and phosphate attached, you'll have a nucleotide. And as those stack on top of each other, the preceding nucleotide will attach to that 3' carbon down there. Phosphodiester bonds are what are connecting the sugar and the phosphate groups. So, like I said, if you have a base and a sugar, you'll have a nucleoside, but if you have all three, you'll have a nucleotide, and these are what stack on top of each other to make those strands. So they connect 3' to 5' ends, so the 3' end of one nucleotide connects the 5' end of the other, but it's always running 5' to 3', it's just the directions are switched on each side. There are hydrophobic interactions between the bases that are driving the double helix. This maximizes and stabilizes those interactions, which is why it's in that structure. DNA has major and minor grooves. In the space filling model, you can see that a minor groove is where more space has been taken up, and a major groove is where less has been taken up, so there's a bigger divot. DNA also has a direction or a handedness. In order to figure this out, I use a staircase technique. I imagine that backbone as a handrail, and which hand would I use to get up there. So this guy is going to go up right-handed, and he makes it, so yay. But if he tries to go up left-handed, unfortunately, he is not happy. So most DNA or bDNA is right-handed, but there are other forms, which include your A form and your Z form. Z form is left-handed, and A form is more compact. But most of the time, you're going to be dealing with bDNA, which is why in the news, if you see a left-handed DNA, you know that they're probably wrong, and you should, I don't know, maybe not read that. But I've got all these guys set to go left-handed, and they make it all the way to the top, so yay, we know it's left-handed. And I've picked up a couple other images that you could practice on. The one on the right looks a little funky, but I thought it was left-handed. So here's my sources, and I'll also provide these in the description. Thanks for listening.